I'm very honored and excited to present our technology and assets in BioKorea 2021. To introduce myself, I'm Songjin Park, CEO of Onegene Biotechnology. Onegene Biotechnology is an early-stage biotech that specializes in developing tri- and tetra-specific drugs, especially for complex diseases such as NASH, fibrosis, and cancer. So those are multifaceted diseases in which the pathogenesis is usually dependent on set of biochemical events and several bioreceptors operating in a companion manner. So we believe simultaneous targeting of those multiple interlinked players in the microenvironment will make a big difference in treating patients suffering from those complex diseases, often called incurable. So to do that, we have developed a dedicated multispecific platform called Unistack, which allows developing protein drugs harboring 3 or more ligands. Since Onegene Biotechnology has been established in January 2016, we have been focusing on developing Unistack, our platform technology. Then we started developing 2 NASH assets from 2019, and from 2020, we attracted a $3.6 million US dollar in our pre-Series A funding, and $12 million US dollar in Series A funding in 2021. So far, we have registered 2 patents regarding our Unistack platform and successfully performing POC for the Unistack technology, and it's first 2 NASH assets so far. So we're at a very exciting stage now. To briefly introduce our management team, I'm Songjin Park, CEO of Onegene Biotechnology. I'm a chemist, and I did my management training in Onegene Group. Onegene Group is a 38 years old auto parts manufacturer whose main customers are Hyundai, Kia, and Ford. I'm a founder of Onegene Biotechnology and an inventor of Unistack technology. Taesung previously worked in major Korean biopharma companies such as Samsung Bioepis, Yuhan, Hanmi, Green Cross for 15 years before joining Onegene. He led and oversaw the CMC of a total 9 pipelines, and 3 of them went to EMA and FDA approval. So he's an expert in CMC. And Josh is a finance expert with experience in IB, security company analyst, and M&A and IR. And he's in charge of value of strategy and IR of Onegene Biotechnology. We have a total of 41 people at this moment with the 12 teams. The team managers will have around 10 years of previous experience in the major Korean biopharmas. So I can assure we have outstanding development capability, although we are very young. And we are currently implementing even QMS so that we can secure data integrity from the development stage. So what is the Unistack? So next I will go over our Unistack platform dedicated in developing multispecific protein drugs with more than 3 targets. So with more ligands, new challenges must be addressed, such as developability, design flexibility, and combinatorial convenience. More specifically, the multi-target drug must be stable, non-immunogenic, and able to harbor any modalities, not limited to only binders. So that we can make a multi-target drug with agonist, antagonist type of signaling molecules, or even enzymes. So, and it must be really easy to make different combinations, so that we can test the best amount of this combinatorial space. So we carefully engineered this Unistack technology to meet such criteria we set. The biggest difference of Unistack is that we make 2 proteins separately and combine them into a single molecule using highly efficient enzyme-mediated conjugation. So that's the key difference. And the thing is that once the large part is optimized, the pipeline expansion is the matter of a 20 to 30 kilodalton protein production optimization. So this allows the true plug-and-play type of a development, which accelerates the pipeline expansions with different composite mechanisms of actions and design flexibility. So this also allows a high flexibility in designing the small part protein structure, which potentially opens new design space for protein design, protein therapeutics design. Structurally, the unit-stack linker serves as a rigid scaffold, providing very spacious room for the ligands compared to the other platforms. So this is more important for agonist-antagonist type of signaling molecules, which has a lower binding affinity compared to the binders. Those type of ligands are more sensitive to steric hindrance and the thermodynamic penalties. So it's really hard to make a multi-target drug with those type of APIs. So so all the parts of unit stack structure is from human serum protein. So we have confirmed that in our in silico and in vitro study that the immunogenicity of all of the pipelines are as low as bevacizumab. And also we found out that the TM of our pipelines is usually high 60s or low 70s degrees Celsius. So it's very stable thinking about this complex structure. The conjugation yield of all our pipelines under development have shown to be greater than like 95%, no matter what protein we chose to conjugate. So therefore, it is a very universal conjugation strategy. And this is a strong backup evidence to say yes when we ask whether unit-stack conjugation is feasible in terms of CMC. After confirming the feasibility of our platform, we chose our first target disease as NASH. I will introduce the candidate coded OGB-21502. NASH is the fastest growing cause of hepatocellular carcinoma and liver transplantation. Presently, there are no treatment approved yet, and many people are suffered from this disease. The unique feature of NASH pathophysiology is that there are multiple causes of the disease in the disease continuum. So, as fat accumulates in liver, hepatic stress occurs, which eventually lead to inflammation, fibrosis, cirrhosis, and HCC. The greatest unmet need is improving liver fibrosis, and there are two types of targets for fibrosis resolution. The first one is remove the liver fat and improve fat metabolism, which are the original cause of fibrosis. Therefore, it's an indirect way of fibrosis alleviation. So second is directly targeting inflammation and fibrosis mechanism, such as collagen synthesis, activation of hepatic stellate cell and Kupffer cell to directly stop and reverse fibrosis. And several past trials that targets only the some part of this whole picture didn't provide a fully satisfying result yet. The year 2020 and 21 have been what's called the year of the bright future, and the clinical results of the drugs that alleviates both steatohepatitis and liver fibrosis finally came out. Among those, FGF21-based drugs showed very promising results based on the efficacy and their safety profile. Especially, the most recent result was in Akero's phase 2A cohort C study with the F4 fibrosis patient. So that's compensated cirrhosis, where fibrosis and inflammation were both alleviated. So this is the first case where we saw this improvement happen in those two group of patients. And So the company asserted that FGF21 has both indirect antifibrotic effect, such as glycemic control and steatohepatitis alleviation, and also has a direct effect in the fibrosis resolution. We also agree with them and expect that the FGF21 may potentially become one of the most promising backbone mechanisms in resolving NASH and liver fibrosis. There are currently multiple FGF21 derivatives under development, and the key question will be which FGF21 program can increase the response rate in serious fibrosis or NASH liver. If you see the NASH landscape of biologics pipelines, there's a clear trend targeting multiple aspects rather than single targets. We totally agree with this direction, considering the complexity of the disease, and to solve. In this context, we first brought this tetra-specific drug using our Unistec technology. So we think that is very natural trend when thinking about the complexity of the liver fibrosis microenvironment. So this is the overly simplified view of the liver fibrosis microenvironment. Hepatic cell, Kupffer cell, and stellate cell are in complex network of communication using cytokines and many other cues to develop and sustain this aberrant repair process or fibrosis. So actually, this really reminds of the complexity level of the cancer microenvironment. So in the cancer field, there are more than already 50 bispecific antibodies and 3 trispecific antibodies exist in the clinical trial. So again, to us, multi-targeting trend in NASH field seems very natural and reasonable. And we really want to accelerate this progress of the field as a company. So our concept is to bring the most effective FGF21 derivative by combining well-defined mechanisms of actions in the aspect of indirect and direct fibrosis targets. To do that, we attach the GLP-GCG incretins to strengthen the indirect antifibrotic effect, and the direct antifibrotic effect is strengthened by adding a cytokine target. So as such, we aim the OGB-2152 will be the quickest and the strongest in improving inflammation and fibrosis in serious fibrotic NASH liver. So do we really need that many targets to have a better efficacy in a fibrosis resolution? The answer is yes, because these are some of our in vitro data showing that having extra targets helps cooling down Kupffer cell and hepatic cell under stress at the cellular level, even at the cellular level. And I think this will be more obvious when we go into the microenvironment of the disease. So, and this is next up is in vivo efficacy data using OGB-2152. So this is an in vitro data plan, study plan for OGB-2152. In the upcoming slides, we will show you our pre-test results, and the main study timeline is seen as in the table. We found out that our 4 ligands behave similarly in the mouse PK. And we also found out that the half-life was 3 to 60 hours in a monkey. Therefore, potentially we can aim weekly dosing for human. We confirmed that the OGB-2152 strongly improved the lipid metabolism and liver functions using MCD and DIO model. The MCD model is to determine how effective the drug is for fatty liver and steatohepatitis resolution. So all mice treated with the OGB-2152 resulted in non-NASH or steatohepatitis free. The difference in NASH score from the control was 3.4 versus 1.1. Of course, histopathologically, we confirmed that OGB-2152 improved the NASH score in MCD and DIO mouse model. And Most importantly, we found out that the OGB-21502 improved the liver fibrosis in CCL4-induced fibrosis mouse model. CCL4-induced mouse model results in a healthy, the harshly induced fibrosis liver, and OGB-21502 treatment prevented cirrhosis progression from 80% of the mouse. And the fibrosis score of OGB-21502 treated group was 2.4 compared to the control 4.0. And this slide shows the histopathological comparison. Compared to the negative control, OGB-21502 has almost no fat accumulation, and there was an improvement in the collagen accumulation and inflammatory cell deposition. And this is very exciting result that all our IPA worked at this target mechanism, such as fat reduction and fibrosis resolution. This is exciting because if you think about this, all targets have different binding affinities, and still they do their own work in the microenvironment, such as defatting and the fibrosis resolution. And using Unistack, we plan to make different combinations of clinically validated targets to develop 4 pipelines. So each combinations are well-validated targets in other fibrotic diseases at the level of clinical stage, or at least at the level of preclinical stage. So we are looking for a partner to explore this whole possible space of a composite mechanism of action together. Regarding OGB-21502, in vivo test result package will be generated using animal models by the end of this year. And the pilot CMC data will be generated in the mid next year. And the GLP test will begin at the end of next year. Next, I will talk about our tri-specific TME targeting immune activator program, which we are seeking for co-development partner too. So a large body of evidence suggests that the tumor creates an immune suppressive microenvironment that inhibits the immune system's natural ability to recognize and destroy tumor cells. Immunotherapy is very strong, but works only in a relatively small fraction of patients. So the big unmet need is how to increase the response rate of immunotherapy. To solve this problem, targeting both the tumor microenvironment and immune checkpoint inhibitor is gaining more attention to enhance the drug response rate in the patients. So we thought we could use Unistack technology to deal with this issue. Recently released the phase 1 result of Phase 1 result of PD-1 CTLA-4 bispecific antibody showed a better safety profile compared to the corresponding combination therapy. So therefore, the combination is gaining more, this combination, so PD-1 CTLA-4 is gaining more attention as a promising bispecific immune checkpoint targets for HCC, RCC, melanoma, and NSCLC. So our approach is to attach the TME targets on top of this immune checkpoint target combination. We're trying FC and albumin as a carrier choice, and we'll decide which one is better based on the developability assessment. Regarding OGB-23501, immunization of binder has been completed and the panning is ongoing. So our goal is to begin co-development once in vivo POC data comes out at the end of this year. So summary is that the Onegene develops tri- and tetra-specific drugs for complex diseases like organ fibrosis and cancer. And the Unistack is a dedicated multi-targeting platform with superior design flexibility and developability. OGB-21502 is the first our Unistack drugs that simultaneously targeting hepatocyte stress, Kupffer cell activation, and stellate cell activation in NASH liver microenvironment. And OGB-23501 is Ongene's first trispecific TME immune activator. So thank you for listening. And as far as I know, Onegene is the only company focusing on drugs only with 3 targets using dedicated platform. So we believe this simultaneously targeting multiple aspects in the disease microenvironment can make a big difference in treating such complex diseases. Thank you, and please let me know if you have any questions.